Okay, and we continue with another talk on immune-targeted therapies for a target that is expressed on a number of pediatric tumors, and that's of great interest. It's B7H3, and this talk is from Tony Choe, Ph.D. candidate from Johns Hopkins, I believe working with Eric Raab's lab. And the title of the talk is Anti-B7H3 CAR NK, Natural Killer Cells for High-Risk Pediatric Brain Tumors. Thank you.
All right. Thank you so much for giving me the opportunity to talk here about my very early preliminary data. Yeah. Hello, hello, test. All right. And then, hold on. It's just the green button, right? Ah, there we go. Ooh, what happened here? Spoilers. Okay. All right. Hello, everybody. I'm Tony. I'm a second-year PhD student, co-mentored by Drs. Charles Bonifant and Eric Raab at Johns Hopkins. And I'm here to present to you about my thesis project, very early data that I have for anti-B7H3 CAR NK cells for high-risk pediatric brain tumors. And quick background on pediatric brain tumors. About a quarter of all pediatric and adolescent cancers are brain or CNS cancers, and they're the most common solid malignancy in childhood and adolescence. And it is still the leading cause of pediatric and adolescent cancer-related deaths in the US. Like Dr. Davila has talked about, there has been great progress in the survival rates of leukemia and lymphoma patients, but in the past few decades, the survival rates of brain tumors have largely stagnated, and this is largely due to the lack of treatment advances for these cancers. I'm going to focus on a small subset of brain tumors in the embryonal brain tumor subtypes. And specifically, medulloblastomas are the most common malignant CNS cancers in children and occurs in the cerebellum. And even though there are 4 molecularly accepted subtypes of medulloblastoma, I'm going to focus on group 3 or MYC-amplified medulloblastomas that have the worst outcomes. And they have a 5-year overall survival rate of around 50%. They have a high risk of metastasis to the leptomeninges. And another type of embryonal brain tumor called atypical teratoid rhabdoid tumors, or ATRTs, are the most common malignant CNS tumors in infants less than 3 years old, and they're molecularly identified by biallelic loss of SMARCB1 or rarely SMARCA4. And these mostly occur in the cerebellum, but they can be supratentorial or multifocal, and these have a 5-year survival rate of about 35%. And treatment options for medulloblastomas standardly include surgical resection followed by focal radiation and some sort of chemotherapy. And there's a lot of risk stratification that happens based on the tumor subtype and the patient age. Like, for example, younger patients are excluded from craniospinal radiation because it can have very severe neurotoxic side effects. And ATRTs, even though there isn't an accepted standard of care, surgery, radiation, and chemo are all considered on a patient-by-patient basis. This is very challenging because the typical age of diagnosis for ATRTs are less than 3 years old, which can exclude a lot of options. And despite the intense treatment regimen these patients receive, as I talked about the survival rates, they have very dismal outcomes and have very high rates of relapse. So this highlights the dire need for safe and effective therapies. I'm going to switch my gears to talk about immunotherapies, because immunotherapies have largely changed the way we treat cancers, and pediatric cancers included. And just going over some currently FDA-approved immunotherapies, we might have heard of chimeric antigen receptor T cells, or CAR T cells for relapsed and refractory B-cell malignancies like leukemias and lymphomas, and also bispecific T-cell engagers for B-cell malignancies. And various monoclonal antibodies, like for CD33 for AML, anti-CD20 for B-cell lymphomas, and anti-GD2 for high-risk neuroblastomas. But currently, there are no FDA-approved immunotherapy for pediatric CNS tumors. So you guys have heard a little bit about CAR T cells throughout the day today anyway, but just repeating a lot of what was already said, CAR T cell treatment for relapsed and refractory B-cell malignancies is very commonly used in the clinic these days for ELL, ALL, and non-Hodgkin's lymphomas, both in adult and pediatric patients, and there's been recent FDA approval for multiple myeloma. And there has been a push in the clinic to bring CAR T-cell therapy to pediatric brain tumors. And here's a couple clinical trials highlighting the GD2 CAR T-cell and also the B7H3 CAR T-cells for DMGs. But there are very widely known limitations of CAR T-cells, one of them namely being cost, of being almost $500,000 US per infusion, and also requiring autologous T-cell procurement and transfer into the patient, and some known toxicity and side effects, including cytokine release syndrome and neurotoxicity. And as traditionally has yielded less than ideal efficacy in solid tumors, a lot to do with some of the reasons named here, antigen escape, proper trafficking to the tumor microenvironment, also the immunosuppressive microenvironment in solid tumors. I'm going to highlight some emerging research in natural killer cell immunotherapy as an alternative to T-cell therapies. There's been some preclinical and clinical work. I'm just showing a few examples of brain tumors and also an AML that our lab is involved in. But what is the advantage of NK cells? They offer a lot of things that T cells don't. Namely, they have an off-the-shelf capability, which means you can actually procure them from a healthy donor for expansion and manufacture. And there's multiple sources of NK cells, including the NK92 cell line, healthy donor peripheral blood, umbilical cord blood, and there has been some effort in iPSCs too. And And when tested in the clinic, these have less risk for graft versus host disease when compared to T cells, and have a much safer toxicity profile when compared to T cells. And NK cells actually have an intrinsic non-antigen specific anti-cancer activity, mostly due to the wide repertoire of activation and inhibitory receptors they have. And they have been tested in the clinic safely in children, namely for neuroblastoma, ALL, and AML. But they do have a few known drawbacks to them. Typically, when delivered systemically, they don't persist too long like CAR T cells do because NK cells don't have a traditional or canonical memory population, and they're much less studied than T cells, and they can suffer from being less potent than T cells, requiring much higher cell doses. But in the clinical world of NK cells, they have been tested pretty successfully in clinical trials. And here's an example of anti-CD19 CAR NK cell done at MD Anderson. It has a very good complete response rate with majority of the patients responding and never reached the maximum tolerated dose, highlighting its favorable safety profile. And also another a phase 1 clinical trial showing the capabilities of a true off-the-shelf product where they have mass expanded and cryopreserved, completely HLA mismatched donor NK cells with no side effects. So going into my research, I really wanted a good validated target for pediatric brain tumors. So there's been a lot of work and validation done on B7H3 as a pediatric brain tumor antigen. Here's some great surface profiling work of known cell lines and patient samples and surface profiling of some known antigens that are in testing right now, including B7H3, GD2, IL-13 receptor alpha, all subgroups of medulloblastoma, ATRT, and also high-grade gliomas. It seems like B7H3 is a very consistently and highly expressed antigen in most of these groups of tumors. And here's some histological data showing the same from a different group where in patient samples, primary patient samples of ATRTs and medulloblastomas, these have very high amounts of expression of B7H3. So what is B7H3? Also known as CD276. It is a ligand that is present on, overexpressed in many solid tumors, as you might have already heard, and its function and receptor are not quite clear yet. And there's a lot of research and clinical trials targeting B7H3 for immunotherapy, including monoclonal antibodies to mediate antibody-dependent cellular cytotoxicity, antibody-drug conjugates, and also CARs. So I was hypothesizing that B7H3 targeted CAR NK cells can show antigen-specific cytotoxicity against human ATRT and group 3 medulloblastoma. So First, I wanted to characterize B7H3 on a few different human ATRT medulloblastoma cell lines, and then develop actual chimeric antigen receptor I will transduce into NK cells, and for testing in vitro and in vivo. So first, I was just doing a quick confirmatory expression by flow cytometry on two different ATR T cell lines, the CHLA-05 and 06, and also a couple different group 3 medulloblastoma cell lines, the DE283 and DE425. And just looking at the surface, it seems like all of these cell lines have very high expression of B7H3. And for the downstream applications, I wanted to generate a B7H3 knockout of these cell lines, so just doing CRISPR-Cas9 knockout into my CHLA-06 ATRT cell line, and then electroporating Cas9 ribonucleoprotein, got very efficient knockout of B7H3, and then followed by bulk and single cell sorting of the B7H3 negative population. And for a chimeric antigen receptor design, wanted to, just a quick background on it, even though you guys have already heard about it throughout the day, the ectodomain, or the single chain variable fragment, SCFV domain, confers you the ability to recognize your target antigen. In my case, it will be B7H3, followed by the hinged transmembrane and co-stimulatory domains. For a lot of CAR T cell products, mostly they're using CD28 or 41BB. Even though 41BB does work pretty efficiently for NK cells, I went with an NK-specific co-stimulatory domain called 2B4. And then also the activation domain, which is same as T cells, CD3 zeta, which is also present on the endodomain of NK cells. I wanted to bring special focus to the SCFV domain of my CAR, because I generated a small library of different affinities for my CAR to B7H3, and this sequences came straight from an already published paper that did a great affinity maturation of the clone of the SCFV that I'm using, and I was able to generate these different affinities for my CAR. And the reason why I did that is because there's been a lot of evidence lately showing that maybe, at least in leukemia studies, a lower affinity or an intermediate affinity actually works better to eradicate tumors rather than a higher affinity. And just as a quick background, a lot of the clinical products, like the CD19 CARs that are used in the clinic, actually have a high affinity. So this is just the workflow of how we isolate and culture primary NK cells. We start with healthy donor peripheral blood, and we follow that with a CD3 depletion to get rid of all the T cells that are in the blood, and then co-culture them with irradiated feeder cells. And in this case, they are K562 chronic myeloid leukemia cell line that are engineered to express 41BB ligand and also membrane-bound IL-21. And after co-culture in these conditions, this selectively expands out the NK cells, and then you can follow that with transduction with the CAR using, we use gamma retroviral vectors. And just an example of the CD3 depletion, on the left you can see pre-depleted whole PBMCs. There's around a 60% CD3 positive population, and post-depletion, we're aiming for less than 1%, but in this case we have around 1.5%, and then we're able to faithfully deplete out the CD3 cells very consistently every time that we do it. And then going on to the transduction efficiency after gamma retroviral vector transduction, I'll follow that with a recombinant protein staining with a secondary stain, his tag stain that has a fluorophore to it, and then just detect by flow cytometry. And I saw that all affinities of my CAR has a very high expression compared to the untransduced controls. And then when testing these in vitro against the CHLA-06 ATRT and also the CHLA-06 B7H3 knockout that I've generated, you can see that at least with comparing the untransduced and the CAR transduced cells, there's antigen-specific cytotoxicity at all effector to target ratios. And I should explain that at higher effector to target ratios, even the untransduced and also the knockout cells do show background killing because these are NK cells and you're going to have some intrinsic killing at a high effector to target ratios. But at least from this one experiment that I've done, this warrants more repeats, but it seems like in the higher affinities when the antigen is present was actually killing better in this short-term cytotoxicity assay, but this remains to be explored more in alternative assays. And a quick preliminary in vivo experiment doing orthotopic xenografts into NSG mice of CHLA-06 ATRT with a firefly luciferase reporter. We can consistently get tumors to engraft with average time to death about 21 days, 22 days. And at time of sacrifice, harvesting the tumor, dissociating into single cell suspensions, I can clearly detect that a big bulk of the tumor mass is still the CHLA-06 ATRT with high levels of B7H3 expression. So in conclusion, human ATRT and group 3 medulloblastomas express high amounts of B7H3 on the cell surface, and these anti-B7H3 CARs can be transduced efficiently into primary human NK cells, and that these cells have antigen-specific cytotoxicity against ATRT in vitro. And these ATRT orthotopic xenografts actually retain the B7H3 surface expression. And currently ongoing and in future directions, we're doing our first in vivo efficacy experiment with the CAR transduced NK cells and the CHLA-06 xenograft. And then also exploring the scFv affinities in other assays, like, for example, serial stimulation for long-term cytotoxicity and also development of the medulloblastoma model in a similar manner of the ATRT model. But you can't talk about immunotherapies really without addressing the tumor microenvironment in solid tumors. And solid tumors in general and brain tumors too, they have a very well-known, well-appreciated tumor microenvironment that is largely stroma and also immune cells that can directly antagonize the effect of immunotherapies. So to combat that, previous work done by Powell Lab and Slusher Lab here at Johns Hopkins have shown that glutamine antagonism is a really great way to remodel the tumor microenvironment, especially the myeloid compartment of it, and kind of induce a stress response in cancer while activating CD8 T cells response against it. So I was thinking, for my future work, I want to incorporate a immunocompetent model in my NK cell work that also comes with optimizing protocols for ex vivo expansion and transduction of murine NK cells, and also establishing and optimizing syngeneic graft models of group 3 medulloblastoma and also ATRT. I want to thank both my labs and also the pathology department and the pathobiology program, which I'm part of, for their support. And I'll take any questions now.
Hi, Nico Josa here. Great talk. Thanks so much for giving us that recap. Question about the glutamine antagonism part of the story and tackling the myeloid compartment. Do you guys have any data, or is it known what is the myeloid landscape of these ATRT tumors in humans or in your animal model?
Yeah. So I think as far as the tumor microenvironment goes, is what very well more studied in gliomas and high-grade gliomas, and is pretty known that in high-grade gliomas, a bulk of the tumor is made up of myeloid, immunosuppressive myeloid cells. But looking at what is present in ATRTs and medulloblastomas, they don't really have as much of a myeloid compartment, but some subgroups and some types have lymphocytic infiltration, also some myeloid in there, but it's very less characterized when compared to gliomas.
Mm-hmm. I hear you. And when you get the glutamine antagonism, what compartment is exactly the one that you guys are thinking of reprogramming?
Well, yeah, the work that I was showing before that was done at Hopkins, they were actually, it's kind of like a three-way approach where the because glutamine is the most abundant free amino acid and that's used as a metabolite for a lot of cellular processes, as cancer cells themselves seem to be starved off by it and induces a stress response, whereas the myeloid compartment turns more into a M1 inflammatory phenotype, even though a lot of immunologists hate the terms M1 and M2, but more of a more receptive phenotype when compared to the immunosuppressive microenvironment, and also kind of stimulating the CD8 cytotoxic T cell response, where they can actually compensate for the loss of glutamine and just through other pathways and become active towards the tumor.
Great, thank you. And then last question, and this is also extrapolating to other brain tumors and CAR T cells. What are the latest thoughts or data showing the proper trafficking of those CAR T cells into those tumors?
Yeah, that's a great question. So in some of the clinical data, clinical titles that I just showed you, in the Stanford GD2 CAR T trial, they actually started off with doing systemic infusion of the CAR T cells. And even though they do traffic to the brain at certain levels, and it does have some sort of effect, intracranial injections just work better. So for all of my experiments personally, I'm just sticking with direct intracranial injections. Mm-hmm.
Thank you.
Yep.
I have a really lovely talk. I have a question about NK cells in the brain.
Yep,
Are they present in certain areas? Are they abundant? And is there a difference in tumor?
I would say normal brain, if you're just talking about the normal brain,
Yes.
I think NKs are definitely not really as present without talking about like infections or cancer or anything. And I think there is a relative normal amount of T cells that are present in the brain, but in the context of a tumor microenvironment, a lot of the times when you're talking about infiltrating NK cells, you think of them similar to the roles that T cells have. They are either trying to be active to the cancer or they are active towards the cancer. And some studies have shown in other solid tumors, I can't really name which ones, that have shown NK infiltration is correlated with better prognosis, but then that's not always going to be the case. But in the terms of brain tumors, I'm not really sure if there's any studies about that with NK infiltrates into brain tumors naturally, but yeah.
And my second question is B7H3 function. So B7H3, I don't know if you found it expressed in the tumor microenvironment, like tumor-associated endothelium or stromal cells at all?
Yeah, that's a great question because I've only seen on the tumor itself, and it is expressed at various low levels and all the way through, like throughout the body too. But strangely enough, even when targeted systemically, it doesn't seem to have any off-tumor effects. But as far as like its expression in the tumor stroma and the microenvironment, I'm actually not sure about that. Yeah.
Yeah. So Brad St. Claude, just so you know, he found B7H3 originally as differentially expressed in endothelium associated with tumor compared to normal endothelium. When he did that screen, the B7H3 came up as a hit. So
Yeah.
it would be interesting to look at that. I don't think it would be a bad thing to target necessarily. It's not a reason not to use that as a target, but it would be interesting because it's quite differential than normal biology, as you said, which is more low level.
Yeah, because at least as far as I'm aware, it's just the antigen density and just the level that is expressed on the tumor is way higher than just any other parts. So.
Thank you.